Now we go back to memory. And the next speaker is Mark Mayford. He's a professor. He works on learning and memory, so crosses both, and uses a mouse model primarily. He uses a range of approaches that include molecular, genetic, and optical techniques, which optical techniques we've not heard much about so far in any of the presentations. His talk today is entitled The Search for a Fear Memory Engram. And I'm looking forward to hearing from you, so thank you, Mark.
Okay, well, thanks. It's really great to be here. I haven't been in the psychiatry department long enough to offer much history. So this first slide is actually an introduction to my talk. Now, for all the clinicians here, this is not an available approved device, but it actually illustrates from an early science fiction series, which you may recognize, Star Trek. What's happening in this image is that a memory is being implanted into the doctor here by direct, presumably, manipulation of neurons in the brain. And that is actually going to be the sort of goal, pattern, outline of what we're trying to achieve in my lab. Now, we're not doing it in people. We're doing it in mice. And we're not doing it with devices like that. So the first question we have to ask is, how do we know what we know about the brain? Okay, how do we know, how does the brain actually represent complex information? And as neuroscientists, we have basically two ways of asking this question, right? One is we do something to the brain. And Larry summarized very nicely his work on how we know about the anatomical structure of regions important for memory by looking at lesion patients, lesion models. So doing something in the brain and seeing a behavioral effect. And the other way we know what might be going on in the brain is by looking at activity. And we haven't looked at I don't think we've looked at it that many talks with that, but people who use fMRI, that's one way of looking at activity at a sort of a broad level. People who implant electrodes can see much more detailed neural firing patterns. And my favorite example, you can find multiple examples of this, but all right, so this is a study from Yitzhak Fried's lab from 2005 where they have electrodes implanted in epilepsy patients waiting surgery, and they show them tons of pictures. Okay, that's not all that important. What is important is that they find neurons, and this just illustrates some of the kinds of exquisite firing properties you can get out of neurons, that seem to fire in response to specific individuals. So in this case, it fires in response to this actress, Halle Berry. And what I found interesting about this paper, people have seen these kinds of things before. I think this was the first paper where they found a neuron that also fired to the letter string for this individual. Okay, so that, you look at a neuron like that, and your first response is, well, that's got to have something to do with your ability to remember or recognize or represent this actress, Halle Berry. Okay, so that's what you think, but there's actually no way to go in and directly test that. So the thought experiment, or the goal of my lab, is to ask, can we somehow take control over those Halle Berry neurons and go in and do the kind of experiment where we can directly manipulate them? So what would happen if you could actually artificially fire just those neurons? Would an image of Halle Berry appear before your eyes? Would you have an hallucination of Halle Berry? Would you think of her name as a sort of memory? How would you recognize that? Okay, so we're not doing that in humans, but we did come up with a way to, oops, wrong way, all right, I'll get used to it, to do that in mice. And the technology isn't all that important, but suffice it to say there are genes that respond to electrical activity, so whether a neuron fires or not. And we've been able to make genetically manipulated mice that basically take this kind of activity response in a gene and use it to express any other gene we want in the pattern of neurons that are active in a time frame of a few hours. In response to natural environmental stimuli. Okay, so now we're in a situation where we can basically show the animal something, label with, say, channel rhodopsin, which most of you probably know what that is. It's this molecule that allows you to, with light, artificially stimulate neurons that express it. Okay, and when we do that, it essentially allows us to do that Halle Berry experiment, to say, take control over a group of neurons, fire them, and see whether it has any behavioral effect on the animal. And I won't go through this because I wanted to talk about some newer stuff, but suffice it to say that there are a series of papers from our lab, from the Tonegawa lab, using this technology to basically show that if you artificially fire a group of neurons, so in an experiment sort of like this, you show the animal something. I'll just stick with Halle Berry, although of course we don't use that in the mouse. And if we naturally label the group of neurons that are activated by that stimulus, and then we put channel rhodopsin into those cells, when you subsequently go back and artificially stimulate them, you can kind of convince yourself that you're producing the percept of Halle Berry, or the stimulus in the mouse. So that tells us something about how the structure of external representations, I like to call this a representation, Susumu likes to call it an engram. I view it as the way the neurons are representing the external world in their firing pattern. Okay. But that's not the question I wanted to just talk about today, because it doesn't tell us what we're kind of have really been interested in for a long time in memory research, which is where do the changes occur? So if we just stick with the Halle Berry idea, I would call the engram where the changes with learning occur that underlie the behavioral change. So you might imagine if you've never seen this person before, you're shown a picture of her, that's going to activate a whole pattern and pathway of neural activity in the processing of this sensory information, and the output is going to be, I don't know who this is. And then if you tell the person this is Halle Berry, blah, 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 you've had many experiences of knowing who this individual is. The next time you see the same external stimulus, the pathway is going to somehow lead to an output where you say, oh yeah, that's Halle Berry. And what we're really interested in when we say the engram, at least to my mind, is how this pathway gets converted to this pathway, with the assumption being this is a recognition area. And where is this sort of needle in the haystack? Okay. And how do we find that? And how do we know if we found that? So I would outline a set of criteria that really were outlined originally by Richard Morris, and we kind of revisited that in a review of how we would know whether we had found this underlying physical change that ultimately underlies a given memory, right? So there is no such thing as the engram. You would have to have a particular task, and you would ask where the change occurs that mediates the behavioral change for that specific task. Well, anyway, how would you know whether you would find, how would you find that? And the three most important criteria are you should identify it. You should see the change with the learning. And these are sort of just standard in all of experimental biology. You should be able to block that change and block or reverse the memory. Here is kind of a crazy one that I'm actually going to show you we have some evidence we can do, which is if you knew exactly what the changes were, sort of like in that first picture where you're just beaming a memory into somebody's brain, in theory, Morris suggested that you could go in, produce those changes, and then get a de novo associative memory without the actual training. Okay, can we do that? And then finally, I would add that as I outlined in that circuit picture, you would like to understand how those little changes in neural connectivity actually control the entire pathway to give you an interesting, the altered behavioral output. All right, so we're going to try to do that in the mouse. I probably don't need to go through this. We're going to use simple Pavlovian fear conditioning, where we're looking primarily at context fear. Basically, do they remember the box that they were shocked in? And this is probably the most studied in the learning and memory field rodent behavioral model. We know a lot about the circuitry involved in this. And so where do we look first for criteria one? Where can we identify a change? And we know that the amygdala is important for fear conditioning. We know something about the inputs to the amygdala, particularly for the tone component. And there was some evidence for criteria one way back in 1997, where essentially, this is from a Shennick-Gallagher paper, essentially what you find is that when you do the conditioning, and then you sack the animal, and you go in and just poke a neuron randomly, and you ask, how strong are the connections of the inputs onto that neuron? What they found is that when you fear condition the animals, you got this increase. And in the unpaired groups and various control groups, you didn't. So that's identifying a change. There's a synaptic change with associative learning. And this paper always kind of bothered me. And not that I didn't believe it, other people have repeated it, but it bothered me conceptually because you shouldn't see that, right? Memory should be sparse. How do I learn the next tone if I just randomly poke a cell and every one of them has undergone potentiation? So we decided to revisit this question with this FOSS-based technology and ask, what would we find if we just looked at cells that were activated with the learning? Okay, so now we're going to fear condition animals. We're going to put a green marker in them, and now we're going to record in the BLA how big are those synaptic responses. So just the same experiment done over, but now we're looking just at the neurons that were activated by the learning. And when we do that, we find a learning-induced potentiation, but now it's limited to the active neurons. Okay, so if we just look at an animal that had nothing done to them, it's going to have some green neurons in the amygdala. They were FOSS active, they had a burst of activity, but they have no potentiation of synaptic inputs. When we looked at the trained animals, we saw this nice increase in response, but then we went and unpaired the tone and the shock, and we also saw the same increase. Now that's a little bit different. And so we thought, well, another thing that always bothered us about those old papers is that while they are learning, they're not learning about the tone when you unpair the tone and the shock. They're always learning about the box they were shocked in. And so we only lost this potentiation when we unpaired the tone and the context. So we actually believe we've found a group of neurons that maybe, at least for criteria 1, we've identified a change that seems to be associated with context learning. And we can show that this lasts for up to 7 days. It's actually reversed with extinction. Well, I guess the increase. And so we've kind of convinced ourselves we've seen criteria 1. So criteria 2, does blocking the change block or reverse the learning? And I'm not going to go through how we do this, but through some molecular manipulations of a molecule involved with LTP, we can get a reversal. So if we virally now deliver only into active neurons this molecule, it's a mutant of a kinase, we will get a reversal of the learning-induced potentiation, right? So we can measure that electrophysiologically, and then we can look behaviorally. And what we find is, so right after we train the animals, we get good learning. So this is just measured of freezing, how much, that's how we quantify their behavioral memory. And right after learning, we haven't had time for the gene expression to occur, so that's our baseline. And then in the animals that express this reversal of the potentiation, we get a loss of the behavioral performance when we get sufficient expression. So we consider that sort of criteria 2. There are better ways of doing it, and we're going to follow up on different ways of doing that. How do we do criteria 3? How do we make a memory where none existed before? And we're not going to exactly do that. But since this is a Pavlovian task, we do have a way of labeling a group of neurons that respond to the US and labeling a group of neurons that respond to the CS, the box. And we can do it in an unpaired way so that the animals actually haven't formed an associative memory. But what we can do is we can put channel rhodopsin into all of those groups. And the way we're going to do that is we don't want to do it just in the amygdala, because our model is that you're carrying information about the context into the amygdala, and that changes at those synapses might be the important thing for the memory, and that's what we're trying to test. So how do we get all of those cortical inputs, which are not really sorted out that well, carrying this information about the box into the amygdala? Well, it turns out that there's a form of AAV virus that is retrogradely transported, okay? So now we inject an AAV that will be retrogradely transported to the upstream areas that input onto the amygdala. And so you can see here the labeling, and this is entorhinal cortex, perirhinal cortex, PFC. So some of the known areas of input to the BLA. And this virus is delivered in such a way that we're only going to label the active neurons, active with CS or with US. So this is very complicated on the top. Suffice it to say that box C is a control box. Box B is the CS neurons that we've tagged, and the US neurons are also tagged. And we get the expression of channel rhodopsin, we wait a while, and now we look at the memory before optical stimulation, okay? So there is no memory, right? There's no significant freezing here because we've delivered them in an unpaired manner. And these are various control groups where we did CS only, US only. Anyway, this is the experimental group where we tagged both the CS and the US, and before stimulation, after stimulation, at high frequency, just in the amygdala, to produce an optical LTP, we produce a apparent fear association to the natural box. I see my red light, so I'm going to finish. We can reverse that fear memory by doing a stimulation protocol that will produce long-term depression. So we can turn it up and down, a natural memory produced artificially by stimulating the CS and US ensembles. Okay, so we think that kind of comes close to reaching criteria 3 for saying that there is an engram in these groups of cells in the BLA. So we would argue that context fear memory is mediated directly by a learning-induced potentiation of CS inputs onto US responsive BLA neurons. I didn't show you some data where we tried to sort out the CS and US responsive neurons. I actually didn't show you this, but the US activated neurons in the amygdala selectively project to a known fear output. That kind of addresses part 4, is how does this change the circuitry? And interestingly, this results actually imply a highly localized rather than a distributed engram for this task. So this task is known to involve the hippocampus, medial temporal lobe structures. We think we can get it just by potentiating inputs into the amygdala. Okay, I'm 2 minutes over. I will thank the students in the lab. This is the work I talk about is most of the work of Leo Cardozo, and we had a visiting professor, Chi Yuan, from Memorial University, who taught us and did a lot of the electrophysiology that I talked about. So, thanks.
Fascinating. I don't know if you know this, but in California, after you reach 70, every 5 years you have to take the computer exam. And gee, the next time I have to take it, which is only 5 years from now, I'm hoping that maybe you'll be able to just implant the answers to the questions in my mind. No, it's just elegant work. Of course it's elegant, and thank you so much for presenting it.